My name is James Butcher, and I'll be presenting an update on our Defeat Duchenne funded research project today on behalf of Alliance Stintzi. Our project's goal is to characterize the role of microbes in muscle health to identify dietary fibers that could potentially improve outcomes in people with DMD. I'd first like to talk about some recent findings from a pilot study which we published last year using the MDX mouse model for DMD. We took microbiomes from wild-type mice and transferred them into the MDX model. We also took microbiomes from the MDX model and transferred them into the wild-type. Controls, we have wild-type mice that received wild-type microbiomes and MDX mice that received MDX microbiomes. And we followed these mice over time, and we were interested in what happened to their muscle health. Now, I don't have time to talk about all of the results today, but one thing I would like to talk about is mouse grip strength. The MDX mice receiving MDX microbiomes got weaker as expected, whereas the wild-type mice getting wild-type microbiomes got stronger. One thing that was interesting was that the MDX mice receiving the wild-type microbiome did not show the same decrease in strength over time. And also the wild-type mouse receiving the MDX microbiome actually got weaker. So this suggests that the microbes that we were transferring were having direct impacts on muscle health. However, there's still a few things that we're not sure about. So one thing is that the MDX model has relatively mild disease. So we want to know whether or not this is also applicable in more severe disease conditions. Secondly, because this was a pilot study, we were not able to measure exactly what was responsible for these changes in muscle health. To begin answering these questions, we are redoing the previous FMT work with a more severe model called the D2-MDX model to identify these muscle modulating features. We looked at the microbiomes of these more severe mice. And these are these preliminary results. And each point represents a different mouse microbiome at a different time. And what we found was that the more severe mice are very different in microbiome composition as compared to wild type. But also, the mouse's microbiome tends to cluster together, as seen with these colors. Just that each mouse has its own unique microbiome, and this could make it difficult for us to identify changes that are happening over time. We pooled the mouse microbiomes from the same time points together and redid this microbiome profiling. What we found when we did this was that we still maintained the large separation between the two genotypes, but now we have very clear changes that we can see over time. So this will allow us to identify specific microbes that are important at early, mid, and late stages of disease. We're currently doing those cross-genotype FMTs with those pooled samples, and with the goal to identify microbial features which are directly impacting muscle health over time. Once we've identified what these are, we will be looking at different prebiotics that we could use to influence these features and improve muscle health. We will be characterizing the microbiomes from people with DMD, and to ensure that these features that we've identified are also present in human patients. In conclusion, we're very grateful for the support shown by Defeat Duchenne Canada, and we're looking forward to presenting updates on this project on the 31st edition.